Perfect. Well, thank you everyone so much for joining us for our first virtual lab notes session of 2021. We're very excited to be here today, and on behalf of the College of Sciences, I want to welcome you. Thanks for joining us. If this is your first time, welcome. And if you've joined us for several of these, we're glad to have you back. So thank you so much. My name is Corey Nelson. I'm the Director of Development in the College of Sciences, and I work on putting these together with some of our wonderful faculty members and researchers. Before we get going, just a few reminders. Make sure that you mute your microphone and keep it muted throughout the duration of the presentation. Make sure that that microphone and camera are both in red at the bottom. It helps with the bandwidth, preserving the bandwidth on the presentation. Again, you can utilize the chat function during the presentation to ask any questions. We'll do questions at the end of Dr. Tseng's presentation, and so there should be about 10 or 15 minutes left to go over those. If you have any issues during the presentation, you can reach out in the chat, or you can check out the support link, which is in the chat. I posted it right there, support.google.com/meet. And just a reminder, again, today's presentation is being recorded. So we will be posting this at a later date on the UNLV Sciences website, and also the UNLV College of Sciences YouTube channel, so you can access it anytime after that. Wanted to just, before we get going, give a quick update on next month's lecture, which will be from Dr. Monica Neda, who's a professor in the Department of Mathematical Sciences. She'll be presenting on mathematical modeling. How can we predict fluid flow, which will have a strong tie-in with a lot of the work that our chemists and our biologists and our physicists do on a daily basis. Math is obviously the language of science, and so we're excited to have Dr. Neda present to us in February. So please join us for that. Also, moving on, wanted to welcome Dr. Shan Tseng. We're so excited to have her with us this afternoon. Dr. Tseng is an assistant professor in the School of Life Sciences at UNLV. She completed her BS in biology at the Massachusetts Institute of Technology in 2003, and she received her PhD with Dr. Hironi Funabiki at the Rockefeller University in 2010. She studied eukaryotic chromosome segregation. Did I get that right, Dr. Tseng? Perfect. And Dr. Tseng transitioned to studying Pseudomonas aeruginosa for her postdoctoral studies with Dr. Matthew R. Parsek at the University of Washington. Her postdoc work mainly focused on biofilm formation of this microbe. Dr. Tseng started her faculty position at UNLV in 2016, where her lab focuses on the functions of the extracellular matrix in biofilm formation with the regulation of Pseudomonas aeruginosa. I keep wanting to make that G hard. I don't know why. Enveloped stress response. Her research is funded by the National Institutes of Health, the Cystic Fibrosis Foundation, Nevada INBRI, Nevada Space Grant, and the Water Research Foundation. And in 2020, Dr. Tseng received the American Society for Microbiology Peggy Cotter Award for early career branch members. We're very excited to have Dr. Tseng with us today, and I will turn it over to you.
Great. So I'm just going to present my screen. So you guys should be able to see my presentation. Great. And you guys can hear me?
Yep, we can see you and hear you. And then for those who are watching the presentation, if you hover over the middle and hit pin to screen, it will make the presentation front and center. And go ahead.
Great. So thank you for that introduction, Corey. Due to some slight technical issues, the camera on the computer I'm actually presenting on does not seem to be working. So we're using a second computer to help with this. So for those who haven't pinned and want to see me while we're doing this, I'm somewhat looking at you, so I apologize for looking down a lot since my slides are going to be on a separate computer. In any case, thank you for coming. So today I'll be telling you about bacterial communities, which are called biofilms, and how they impact us as humans. So I want to start by taking a moment and thinking about bacteria. So what do you think of when you think of bacteria? So just take a second, think about that, and type your response in the chat. Anybody? What do you think of? Germs, some people. Overall growth. What else? What do you think of? Infection? Bad, like diseases, okay. So here are some of the responses I've received in the past in regards to this question. So some people think of water-based environments where bacteria are swimming around kind of by themselves in these vast oceans. Other people think about bacteria in our food. So for instance, bacteria that can cause disease, as mentioned. And so, for instance, foodborne pathogens such as salmonella on the raw chicken skin, right? And so here again, you typically think about like individual bacteria kind of scattered all over that chicken skin. Alternatively, you might also think of bacteria in our food that's actually good for us. So for instance, the active live cultures of probiotic bacteria in yogurt. And again, you think of these, in general, I would say people actually think of individual cells kind of all by themselves when they think about bacteria, and they're kind of, you know, individuals in this vast environment. But in reality, most bacteria, up to 80%, live in communities with other microbes. And so these communities are called biofilms. And so instead of actually being free swimming and by themselves, more often than not, bacteria are actually clustered in these aggregates and with other microbes in their local community. So shown here are 3 scanning electron micrographs of bacterial biofilms. So shown on the left is an example from the oceans. You can see here this cluster of cells is the biofilm that's attached to this other marine microbe. This is a diatom, and this relationship is symbiotic. So the bacteria provide vitamins to the diatom, and in exchange, the diatom provides sugars as a source of food for the bacteria. In the middle here is actually a salmonella biofilm on the surface of chicken skin. So you can see that there's lots of bacteria kind of clustered together here. And then on the far right is the biofilm that you can find of probiotic bacteria in yogurt. So these long rods are lactobacilli, and these circle shapes are streptococci in the yogurt. So what you might notice in each one of these images is that there's either goopy material or stringy material kind of between the cells, and this is kind of most obvious in this image where the streptococci have all the stringy material between them. And so this goopy stringy material is the extracellular biofilm matrix, and it's produced by the bacteria to hold the biofilm together and to various surfaces. So for instance, the surface of the diatom or the chicken skin. Since up to 80% of bacteria in biofilms, or in biofilms, then biofilms are all around us. And so here's some examples of really cool and interesting biofilms. So if anybody's ever been to Yellowstone and saw those beautiful colors, these are microbial mats that are biofilms. Biofilms are also used in industry. So for instance, the Southern Nevada Water Authority, as well as water treatment plants across the world, use biofilms to clean our wastewater such that water can then be put back into the environment and reused. Biofilms are also used in agriculture, for instance, for the production of some foods, such as cheese. And in our guts, as well as the guts of all animals, are microbes that live in biofilms. That's important not only for helping us digest our food, but also for keeping harmful bacteria in check. However, not all biofilms are good. So in the environment and in industry, biofilms do what we refer to as fouling. And so these bacterial biofilms form on the surface of things such as ships or industrial pipes, and it leads to deterioration of these surfaces because the microbes actually corrode the metal. And therefore, these biofilms have to be constantly removed to prevent that corrosion, or in the case of pipes, be removed so that it doesn't clog the pipe. Biofilms can also cause disease. An example of this in agriculture is infections of the cow udder. So not only is this painful for the cow, but it makes the milk unusable for us. Biofilms cause disease in us as well. And so an example here is periodontitis, where the biofilm grows between the tooth and the gum. And this leads to inflammation of the gum, then the long term leads to gum damage and deterioration of the underlying bone that support your teeth. So as I showed you on the previous slide, biofilms form on a variety of different surfaces, and this includes the human body. So biotic surfaces of the human body where bacterial biofilms have been shown to form include the teeth, the skin, the heart valves, the sinuses, and the lungs. In addition to these biotic surfaces, abiotic surfaces in the human body, such as the surface of any medically implanted device, can also have biofilms form on them. And so biofilms forming on medically implanted devices is actually one of the main reasons these devices have to be replaced before the lifetime of the device. And this often involves surgery to get these devices in and out. So the problem with biofilms growing in and on the human body is that bacteria that reside in these biofilms can cause infection. Biofilm-based infections are problematic because bacteria in biofilms are more tolerant to antimicrobial compounds than their free-swimming counterparts. So let's say you have an infection, but the doctor doesn't realize it's a biofilm-based infection, right? You go to the doctor, they give you antibiotics, the traditional concentration of antibiotics, to treat that what they think is a free-swimming bacterial infection. While this antibiotic treatment does kill some of the biofilm cells, not all the bacteria will be effectively killed because they have a higher tolerance for this antimicrobial. Therefore, any biofilm bacteria that's not killed can reseed the infection after the antibiotic treatment is over. And because of this, biofilm-based infections are often chronic in nature. Because biofilms are so resilient to antibiotic treatment, such infections cost over $100 billion a year in the US alone to treat. Furthermore, current therapies really target free-swimming bacteria, and we have very few tools to deal with these chronic biofilm-based infections. Mainly, the main way we deal with them is actually physically removing the biofilm, which can be problematic depending on where it's growing. So therefore, biofilms are an important human health problem, and understanding biofilms is critical for the development of better treatments for such infections. And in general, understanding biofilms will allow us to better manipulate them to the benefit of society. And this includes both minimizing the negative effects, such as an infection, and also maximizing their positive effects, such as in the treatment of wastewater. So understanding biofilms is a major focus of my lab. And to study biofilms, my lab uses the organism Pseudomonas aeruginosa. It is a gram-negative bacterium, and this means that it has two membranes in its cell envelope surrounding that cell and defining it from its environment. To study biofilms, I should say this is a pseudocolored electron micrograph. And so each one of these rods is an individual bacterium, and so there's 4 bacteria here. This thin, long structure is called the flagellum, and this allows the bacteria to actually swim in fluids. So P. aeruginosa is one of the major organisms used to study biofilms, and is actually a common microbe that can be found in the natural environment, so such as soil. And pretty much anywhere you find humans around, you can find Pseudomonas aeruginosa in the environment. It is an opportunistic pathogen, meaning that while it does not infect healthy humans, it does infect those with compromised immune response. In terms of its ability to cause disease, P. aeruginosa is one of the top 5 pathogens that causes hospital-acquired infections, with about 51,000 cases a year in the US. In addition to these typically acute hospital-acquired infections, Pseudomonas aeruginosa also causes chronic biofilm-based infections, for instance, in the lungs of people with cystic fibrosis, and these infections in the cystic fibrosis airways can actually last for decades. P. aeruginosa also causes chronic infections in the lungs of those with COPD, in the lungs of those on ventilators, in the foot wounds of people with diabetes, and in the eyes of people who wear contact lenses and do not properly clean them. So here is a 3D rendering of a P. aeruginosa biofilm that we grew in the lab. This is not exactly what you would find in an actual infection, but gives you an idea of what a biofilm would look like. So the bacteria here are expressing a fluorescent protein. This allows us to visualize the cells in the biofilm using a confocal microscope. The surface that the bacteria are attached to is down here, and you can't see it. And so under the conditions that we use to grow this biofilm, the bacteria grow into a biofilm that's very structured in shape. And so in the field, we refer to each one of these aggregates as a mushroom because it kind of looks like that. And this particular mushroom structure is about 100 microns wide and 75 microns tall. And so to give you an idea of scale, each one of this particular mushroom structure contains about 500,000 bacterial cells. And this entire image, you're seeing about 2 to 3 million bacterial cells. So how do bacteria form these microbial communities? So here's a cartoon of biofilm formation. This is the flat surface that the bacteria are going to colonize, and we're looking at it from the edge on, so from the side. So in biofilm formation, first you have individual cells attaching to that surface. That's depicted here by this green rod with this swimming appendage. Once on that surface, the bacteria change their gene expression and suppress their ability to be motile. Instead, they start growing in what we refer to as a sessile lifestyle, where they produce and secrete the material that holds them together, and that's shown here is this darker green shading around the cells. And so this darker green shading represents the extracellular matrix material that I mentioned previously. As this biofilm continues to grow and mature, it's going to get larger, more cells and more matrix materials to hold the biofilm together. And ultimately, at some point, the bacteria in the biofilm will disperse. So they'll leave this aggregate either as individuals or as clusters to colonize new surfaces. And in the case of disease, spread infection. So here we're looking at the side view of that mushroom structure developing. And so you can think about it like a mushroom sliced vertically. So similar to this idea, in terms of how this cartoon relates to that 3D image I showed you on the previous slide. Here are a few of those steps and what they look like under the microscope, so actual bacteria forming biofilms. And so note here that the scale is not the same for these images. The bar in each case is 20 microns. So this image is a lot more zoomed in than that one, per se. So in these images, the green is the P. aeruginosa bacteria, and in red is one of the matrix components that P. aeruginosa makes. And so in these first two images, we're looking top down onto the surface that the bacteria are interacting with. And so that's instead of edge on, as shown here. So you can see that initially during attachment, you get individual bacteria on the surface, and we don't see the motility appendage because it's not labeled. As these cells grow into a microcolony, we see more and more cells piled up on each other. We also start seeing the cells producing and secreting this matrix material that holds the biofilm together, and again, one of those is shown in red. This far right image is the fully mature biofilm, and instead of looking down from the top, we're now looking from the side. So similar idea to this, sliced mushroom / cartoon image. And so the bottom, the surface with which this biofilm is attached is on the bottom. And so what you notice here is that this specific matrix component kind of surrounds the outside of the cells in this mushroom aggregate, as you can see here in the red. In addition, this yellow is actually a combination of the green bacteria with the red matrix material overlaid on top of it, which is why it shows up as yellow. And so in addition to being surrounding the outside of this aggregate, you can see that the matrix component is also at the base of this biofilm where it's attached to the surface. And so you might notice that there's none of the matrix component, this red matrix component, in the middle of the biofilm aggregate. So it does not actually, this specific matrix component is not there, but there are other matrix components there. They're just not visualized here. So what is the matrix composed of? And to describe this, I just want to take a step back and mention that biofilms are pretty much defined as the cells plus the matrix. So shown here is a cartoon that will not be to scale, but the cells are shown as green rods. And so the first major component of the biofilm matrix material are exopolysaccharides. And so exopolysaccharides are simply chains of sugars, and that's depicted here as the strings of red hexagons. The individual sugars are connected by the cell as it is secreted out of the cell. So these long chains of sugars do not exist inside the cell, they're synthesized extracellularly. The exact identity of the sugar, how they're connected, and the biochemical properties of these exopolysaccharides is specific to the organism. And organisms can also produce multiple exopolysaccharides in their biofilm. So for instance, Pseudomonas aeruginosa has the ability to make 3 different exopolysaccharides. The matrix also contains DNA that's outside of the cell, which is why we refer to it as extracellular DNA, or eDNA for short. And that's depicted here by these black double helices. This DNA is actually the same as what's inside the bacterial cells. And the main way that the DNA gets outside of the bacterial cell in a biofilm is due to cells bursting. And so there is death of some of the bacteria via lysis in the biofilm. That's actually critical for the formation of the biofilm. And this is critical because it does get certain components of the matrix into the extracellular space, such as the eDNA. So interestingly, any DNA can be used by the bacteria in their biofilm. And so, for instance, at the site of infection, any human DNA that gets outside of the cells, like, let's say, an immune cell were to burst, that extracellular human DNA can now be used by the bacteria to make the biofilm stronger. And this is interesting because the immune system actually combats free-swimming bacterial infections by bursting immune cells at the site of infection. And so this is because lots of extracellular DNA is actually toxic to free-swimming bacteria. So while bursting of immune cells, which again is a normal part of the immune response to combat these free-swimming infections, may actually be detrimental in a biofilm-based infection. So the third major component of the biofilm matrix are membrane vesicles, depicted here as this yellow circles. These form in part, again, via the bursting of bacterial cells, but they can also form by having the bacterial cells in the biofilm bleb off parts of its membrane. Because these are vesicles, cargo can actually be inside the vesicle, and in the biofilm, these cargo have been suggested to be important for the communication and sharing of resources between the bacteria in the biofilm. The last major component of the biofilm are the matrix proteins. And so some matrix proteins are individual globular proteins, as depicted here by these blue rectangles. However, other matrix proteins can actually form long polymers, a similar idea to these eDNA and exopolysaccharides. I've just not depicted that here. So proteins inside the cells become extracellular, again, either through lysis of cells or through secretion of those proteins from intact bacterial cells. And so the overarching question of my lab is, what do these matrix components contribute to the biofilm? What functions do these components have? What happens if you were to remove them? And so three main functions that have been suggested for the biofilm matrix are structure, protection, and nutrient storage. So the structure one is kind of obvious because the matrix is critical for holding the biofilm together. So shown here are two examples of how the matrix components play structural roles in P. aeruginosa biofilms. So on the top, on the left, is a wild-type bacterium it's formed a biofilm, and on the right is a mutant that no longer makes one specific matrix protein. It's called CDRA. So in green are the bacterial cells, in red is one of the exopolysaccharides that P. aeruginosa makes. And so we're looking down onto the top of the mushroom structure. And so you see that in wild type, if you're looking top down onto that mushroom structure and looking at the cap of that mushroom, the exopolysaccharide surrounds bacteria that are in the middle of this aggregate. In comparison to this, in the mutant that lacks the ability to make this one specific matrix protein, while the exopolysaccharide is still being made, you still see a little bit of red here, it doesn't stay associated with the biofilm cells. And so the role of this specific matrix protein is to connect this exopolysaccharide to the cells. And this protein is critical for the structure of the biofilm. So in comparison to what's shown up here in terms of a specific matrix protein being necessary to be there to form a biofilm, below is an example of what happens if you were to degrade matrix components. So the green, again, is the bacteria in the biofilm, and again, we're looking top down onto that biofilm. And because of the way these biofilms are grown, instead of getting these mushroom structures that we've been talking about, we actually get a thick mat style biofilm instead. So on the left is just an image of the biofilm. And so on the right is what happens if you take this biofilm and then add large amounts of an enzyme that degrades exopolysaccharides. And so what you see here is that there's very little green bacteria left in this biofilm. And so by adding enzymes that can degrade the exopolysaccharides of the biofilm matrix, we can actually break up the biofilm. So in addition to the structural role, the matrix has also been shown to protect the bacteria. So shown here is an example, again, from P. aeruginosa, based on work I actually did as a postdoc. So we're looking here at the side slice of that mushroom structure, again, not from the top. And so this is wild-type bacteria. The surface that the biofilm is attached to is on the bottom. In this image of the wild-type biofilm, this has been treated with an antibiotic, and we can this antibiotic is commonly used to treat P. aeruginosa infections. We're able to visualize the antibiotic because it's been labeled with a fluorescent molecule. So shown on the left is a merged image with the bacteria in green and the antibiotic in blue. The right is the same image where I've removed the cells so you can more easily see where the antibiotic is. So when the antibiotic comes into contact with this biofilm aggregate, it does not fully penetrate the aggregate. It doesn't get into the middle. And in fact, it just gets stuck around the outside of the structure. And so the biofilm matrix here is serving as a diffusion barrier that binds to the antibiotic, and it prevents the antibiotic from getting into the middle of the aggregate. In this particular case, this occurs because the antibiotic is very positively charged, and the eDNA and the lipid vesicles in the biofilm are very negatively charged. So the antibiotic can interact with these components of the matrix via ionic bonds. And this actually illustrates one of the problems associated with treating biofilm-based infections with traditional antibiotics. So while the cells on the outside of this aggregate that are in contact with the antibiotic might get killed, the cells in the middle are fully protected. And so once antibiotic treatment ceases, the cells in the interior can reseed the infection. So lastly, the matrix has been suggested to serve as a place to store nutrients. This is because the matrix components are made up of the same macromolecules as inside the cell. Therefore, under conditions of starvation, theoretically, the bacterial cells in the biofilm could eat the matrix components to survive. So I've depicted the model of this idea here. So again, this gray is the surface the bacteria are attached to. The green rods are the bacteria. This darker gray shading represents the biofilm structure. So this string of red hexagons, again, is exopolysaccharides. And so remember, the exopolysaccharides are just strings of sugar. These strings are too large for the cell to actually take up, but if the cells are being starved, theoretically, they may secrete an enzyme, shown here as these black Pac-Men, to degrade that exopolysaccharide. This would then release simple sugars into the biofilm, as depicted here as these individual hexagons. And these simple sugars can be taken up by the cells as a source of food. So obviously, the bacteria don't want to make too much of this enzyme, because otherwise you end up with a situation that I showed you here on the left, where you fully degrade your biofilm, and that wouldn't be good. But nonetheless, the biofilm matrix can be used as a place to store nutrients for later use by the microbes. And so here I'm only showing this as a cartoon, because while parts of this model have been shown to be possible, as of yet there's no data to show whether this actually occurs in biofilms or not. So towards understanding the various functions that the matrix contributes to the biofilm community, my lab has been focusing on the matrix proteins, and this is because the potential for different biochemical activities for proteins is much greater for proteins than for DNA, polysaccharides, and membrane vesicles. And so to address this question as to what functions do the matrix proteins provide to the biofilm community, we actually first had to identify the matrix proteins that are there, because this hadn't been done. So using proteomic approaches, we identified 60 candidate matrix proteins in Pseudomonas aeruginosa biofilm matrices. And based on what was known about these proteins in the literature, we categorized them into one of 4 classes based on what we think they might be doing in the biofilm matrix. So 5 proteins look like they might be involved in forming or maintaining the structure of the biofilm. 20 proteins are predicted to be involved in protecting the biofilm. 29 proteins we think may help the biofilm cells acquire nutrients back from the matrix, or may be involved in helping degrade the matrix selectively that would then allow the biofilm to actually grow. And then lastly, there's 6 unknowns, because these proteins haven't been studied as of yet and don't obviously look like other proteins that have been studied. So today I'll be telling you about 2 proteins that we've been studying, OPRF and ecotin. And so we categorized OPRF as potentially playing a role in the structure of the biofilm, because previously published work has shown that it is important for the attachment of P. aeruginosa to human cells. And so we thought, well, perhaps there's also a role of OPRF in the adhesion in biofilms. We categorized ecotin as having a potential protective role, because ecotin had previously been shown to inhibit enzymes that degrade proteins inside and part of cells. Therefore, we thought perhaps ecotin in the matrix may prevent enzymes from degrading the biofilm matrix proteins. So first, we'll talk about OPRF. So as I previously mentioned, P. aeruginosa is a gram-negative bacterium with 2 membranes separating the cell from its environment. So to get molecules such as sugars and ions through these membranes, so they can actually get inside the cell, there are proteins in these membranes that help transport those molecules inside. So OPRF is one of these transport proteins, and it's drawn here as this yellow cylinder. It creates a selective pore in the outer of the 2 membranes of the bacteria. Previous work by other groups showed that OPRF interacts with a protein complex shown here in blue that binds to the surface sugars of the human cells. This protein complex is drawn as 4 ovals put together because it is composed of 4 of the same protein in complex. So this interaction of OPRF with this protein complex and the surface of these host cells is important for the attachment of the bacteria to human cells. Therefore, by removing OPRF, you actually make the bacteria less pathogenic because the bacteria have a reduced ability to attach to the host cells, making them less able to infect. So based on what was known about this interaction of OPRF with this blue protein complex and the ability of this complex to bind to sugars, we hypothesized that OPRF may be involved in the adhesion of components of the biofilm. And so shown here are three options that are not mutually exclusive. So first, perhaps OPRF plays a role in adhering the cells of the biofilm together. So one of these blue protein complexes could interact with multiple OPRF molecules on different bacterial cells, and this may be one way to cross-link the bacterial cells together in the biofilm. In addition, OPRF may help adhere the biofilm matrix to the cells. So here, shown here is the exopolysaccharides. And so OPRF would interact with this blue protein complex, and another part of this protein complex would interact with exopolysaccharides, cross-linking these two components of the biofilm. Lastly, OPRF may be involved in attaching the vesicles to the biofilm matrix, and so through a similar mechanism as shown over here, the only difference here is this lower part now is a vesicle, and this works because OPRF is also in the membranes of these membrane vesicles. And so OPRF may play a role in helping retain these membrane vesicles within the biofilm matrix. So to study the role of OPRF, we first deleted the gene from the bacteria. And so this delta OPRF denotes a mutant that lacks the ability to make this protein. We then tested its effect on biofilm formation using a static standard biofilm model where we put bacteria in a nutrient broth and then into this plastic well. Then we allowed the bacteria to grow. And as the bacteria grow, they're going to form a biofilm at the air-liquid interface. They're also going to attach to that surface of the plastic well. Therefore, when we remove the broth, the bacteria leave this bathtub ring of cells and biofilm attached to that plastic. We can then stain this biomass as shown here in purple, and then quantify the amount of biofilm that's attached to that plastic well. So on the y-axis here is the amount of biofilm, a relative amount of biofilm, and it's normalized against wild type, which is set at 1. And so you can see with the OPRF mutant, it produces 75% less biofilm than wild type. However, we're actually not the first group to look at the effect of OPRF on biofilm formation. Using a similar assay, the Chevalier lab previously published that mutants of OPRF produce significantly more biofilm than wild type, while we see significantly less. So why do these data conflict? After looking at a few things, one thing we noticed was that while we use the standard nutrient broth for this assay, it's called TSB, they used a different one called LB. Because there were differences in the nutrient broth, we want to test if this was the cause of this difference where OPRF mutant causes less biofilm in one case and more in the other. So shown here on the left is the data I just showed you on the previous slide, with the OPRF mutant making 25% the amount of biofilm that wild type does. So when we did this exact same experiment but changed up the nutrient broth to LB, we saw this instead. We saw something closer to what the Chevalier lab saw. While we never see that twofold increase that they report, we consistently see that the OPRF mutant produces statistically similar amounts of biofilm as the wild type when the cells are grown in LB. Therefore, the role of OPRF in biofilm formation is dependent on the conditions under which the bacteria are grown. But why, right? What's different about these two nutrient broths? So staring at the ingredients list, we notice two major differences between them: sodium chloride and glucose. So in the case of sodium chloride, TSB has low concentrations of sodium chloride, while LB has very high concentrations, about twice as much in fact. Glucose was another major difference. So TSB contains glucose, LB does not contain glucose. Therefore, we decided to alter these two components to determine if they're the reason for these different abilities of the OPRF mutant to form biofilms. So we tested these components individually, holding all the other ingredients in these nutrient broths the same. So in the case of sodium chloride, we made TSB with high concentrations, LB with low concentrations. In terms of glucose, we made TSB without glucose, and we made LB with glucose. And so by individually altering these components to look like the other broth, essentially, for that one component, while keeping all the other ingredients the same, we can test if these specific broth ingredients are associated with this condition-dependent effect of OPRF on biofilm formation. So shown here is that same static assay I've been talking about. On the y-axis is the relative amount of biofilm. To declutter this graph, I've actually removed the wild-type bars. So all the bars for wild-type would be set at 1 because the data is normalized against the wild-type grown in the same nutrient broth. And so I've drawn a dashed line at 1 to help remind you where wild-type would be if it were shown. So all the bars here are going to be what happens with the OPRF mutant in these various nutrient broths. And so currently I'm only showing you what happens to the OPRF mutant in TSB, in the standard TSB and LB. So this is similar to what I showed you before, right? So the OPRF mutant produces 25% the amount of biofilm as wild type when it's grown in TSB. You get a slightly higher amount over on this right experiment, but nonetheless, both of these are statistically significantly less than the wild type. In comparison to this, in LB, for the OPRF mutant, we see statistically similar amounts of biofilm as wild type for both of these experiments. So what happens if we were to alter the amount of sodium chloride in these broths? So if we increase the amount of sodium chloride in TSB, we see more biofilm forming. If we lower the sodium chloride concentration in LB to match that of TSB, then the OPRF mutant makes less biofilm. So overall, these results demonstrate that OPRF mutants form less biofilm when they're grown in broth that contains low sodium chloride. So what about the glucose? So if we remove the glucose from TSB to match that of LB, the OPRF mutant produces much more biofilm than in the presence of glucose in TSB, and this is equivalent to the amount that you see statistically in wild type. In addition, if we now add glucose to the LB, and our OPRF mutant now forms less biofilm than wild type. And so these results show that OPRF mutants form less biofilm in the presence of glucose. And so currently we're investigating how the sodium chloride and the glucose in these nutrient broths actually affect the response of this mutant OPRF cells to help understand the mechanism underlying this condition-dependent effect of OPRF on biofilm formation. So stay tuned for more on this story in the future. So this first story I gave you shows you a glimpse as to how matrix proteins are involved in the structure of the biofilm, but how can matrix proteins be involved in protecting the biofilm? So here we're going to transition to talk about the other matrix protein, ecodin. So we predict that ecodin would be involved in protecting the biofilm based on its published function in cells. So ecodin is known to inhibit an enzyme called neutrophil elastase. Neutrophil elastase is a protein that degrades proteins. And so neutrophil elastase is important because it is secreted by immune cells at the sites of infection to actually kill the bacteria. And the way neutrophil elastase does this is by degrading the proteins in the membranes of the bacteria, leading to a reduced structural integrity and death of that microbe. And Pseudomonas aeruginosa commonly comes into contact with neutrophil elastase at sites of infection. So therefore, we hypothesize that ecodin in the biofilm matrix may protect the biofilm from attack by neutrophil elastase during infection. So here's a model of how we think that works. Here are two biofilms, on the left without ecodin, on the right with ecodin. Again, the green rods are the cells, the strings of red hexagons are the exopolysaccharides, the black helices are the eDNA, the blue rectangles are the matrix proteins. So on the left, again, is without ecodin. On the right is with ecodin, and that's depicted here as these blue stars. So in the absence of ecodin on the left, when neutrophil elastase, which I'm going to show here as gray Pac-Man, is secreted by immune cells, it can attack this biofilm. And in the absence of ecodin, neutrophil elastase can lead to the killing of the biofilm cells and degradation of the matrix proteins. And overall, this would lead to a decrease in the integrity of the biofilm. In comparison to this, in a biofilm with ecodin, neutrophil elastase can be inhibited and neutralized by the ecodin. And this would protect the biofilm cells from being killed and the matrix proteins from being degraded. And overall, this leads to preservation of the biofilm integrity. So to test if ecodin can protect the biofilm, We started by deleting the gene from the bacteria, so denoted here as delta eco. These are mutant cells that can no longer make ecotin. We grew biofilms of the wild type and the ecotin mutant. And again, we're looking top down on that biofilm, and based on the way we grew it, we get that thick mat style biofilm. So shown here in green are the living bacteria, and you can see individual purple bacteria here, and those are the bacteria that are actually dead. So you can see that removing ecotin has no effect on the ability of the bacteria to make biofilm. So visually, these are the same, and they have similar levels of dead cells in the biofilm, which is quantified below these images. So to test if a biofilm of an ecotin mutant is more susceptible to neutrophil elastase, we treated these two biofilms once they'd grown up with neutrophil elastase. And so what you see is that for the wild type biofilms, there's very little difference between the neutrophil elastase treated and the sample that's not neutrophil elastase treated, right? So they have similar levels of dead cells in these biofilms. In comparison, for the ecotin mutant, you see significantly more dead cells when the ecotin mutant biofilm has been treated with neutrophil elastase relative to when it's not. Therefore, these results show that ecotin can protect biofilm cells from neutrophil elastase mediated killing. What this result doesn't show is whether, or what doesn't differentiate, I should say, is the effect of the intracellular ecotin, i.e. the ecotin that's inside the cells, versus the extracellular ecotin that is associated with the biofilm matrix outside of the cells. So to examine this, we changed the experiment slightly. And so for this slide, I'm only showing you biofilms of the ecotin mutant. And so right now, what's up is data that's similar to what I showed you on the last slide, where a biofilm of the ecotin mutant is more susceptible to cell death upon neutrophil elastase treatment relative to the same biofilm that's not treated with neutrophil elastase. And so the way we changed the experiment for this data, and I'm going to show you on the right, is that after we grew the biofilms of the ecotin mutant, we added to the biofilm purified ecotin protein, which is denoted here as R-ecotin. After adding the protein and letting it incubate for a little bit, then we washed off any of the purified protein that didn't actually stick to the biofilm. And so what's shown here on the right is actually biofilms where there's no ecotin inside the cells, but there is ecotin outside of the cells. And so we then treated these biofilms with neutrophil elastase. And what we see is that if we add the purified ecotin to the biofilm matrix before we treated with the neutrophil elastase, we were able to protect the bacteria in the biofilm from being killed. So similar to the wild type biofilms I showed you on the previous slide, there's very little difference between the neutrophil elastase treated and untreated samples with the mutant ecotin biofilm that has this purified ecotin added to its matrix. And so overall, these results show that extracellular ecotin can protect biofilm cells from neutrophil elastase mediated killing. And so this was actually all done kind of like a test tube in the lab situation. And so one question we have is, well, can ecotin actually protect biofilm cells in an actual infection? And so we're currently collaborating with scientists at the University of Calgary to address this question in an animal model. So I just want to finish up by summarizing some of the key points. So I started today by telling you about how biofilms are all around us and that they're both beneficial and detrimental to humans in industrial, agricultural, and medical settings. In particular, I focused on the role of the extracellular matrix and its functions to the biofilm community. I gave you two examples from our recent work on how matrix proteins are important to the biofilm. So first, I showed you that OPRF mutants do not form proper biofilms, and this is dependent on the conditions under which the biofilm is grown. Second, I showed you how ecotin in the biofilm matrix can protect the biofilm resident cells from being killed by a protein-degrading enzyme that the immune system produces. But ultimately, you might be thinking to yourself, well, why should the general public care about our work? And so while it's not our goal to develop the applications of our work, what I have shown you today is applicable to society. So in the case of OPRF, there is current work by others focusing on using OPRF to develop a vaccine against Pseudomonas aeruginosa. So such a vaccine would lead to the production of antibodies against OPRF in the human body, and such antibodies are likely going to block the function of this protein for any bacteria that infect a person who's been vaccinated. And so in such a case, in addition to potentially preventing infection of free-swimming Pseudomonas aeruginosa, this vaccine may separately also change the ability of the infecting bacterium to form a biofilm. Whether inhibiting OPRF by such antibodies would actually promote or prevent biofilm infection is actually based on the local conditions, right? Since the OPRF immune had these condition-dependent effects on biofilm formation. And therefore, understanding how the conditions actually affect biofilm formation in an OPRF mutant is important for our understanding of how the OPRF-based vaccines will affect biofilm-based infections of P. aeruginosa. In the case of Ecotin, this is a little bit more obvious. So since we've shown that Ecotin can protect biofilms, perhaps therapeutics can be developed that inhibit the function of Ecotin, and treatment of biofilm-based infections with such a therapeutic would theoretically make the biofilms more susceptible to killing by our own immune response, and therefore make it easier to eradicate such biofilm-based infections. So overall, I hope I've convinced you that basic research into understanding how biofilms form and protect the resident bacteria is important for allowing us to better manipulate them to the benefit of society. So I want to thank the people who did the work. This is a current picture of my lab. Here are the lab members who worked on, or who currently work on Pseudomonas biofilms. Other members of my lab do other aspects of microbiology that I did not have an opportunity to share with you today. We recently started with a collaboration with Cesar Corsagara to look at the role of OPRF on the ultrastructure of individual cells and the biofilm. I started the Ecotin project while I was a postdoc in the lab of Matt Parsek, and this work was done in collaboration with Courtney Reichart, Joe Harrison, and Mike McCoss at that point in time. As I mentioned, we recently started a collaboration with Paul Cubes to look at the role of Ecotin in the biofilm matrix in an actual infection. And we received generous funding for our work from these agencies. And finally, I'd like to thank you for your attention. I'd be happy to take questions.
Thanks so much, Dr. Tseng. That was great. I really appreciate you taking the time out of your day to present for us. There were a couple questions that came in during the presentation. So I'm going to start at the top with, it looks like last question. It was when you were discussing like how the communities are created, how the biofilm communities are created. And the question was, does this get produced naturally? I'm assuming that's the bacteria, the biofilm, or does it get produced in certain conditions? Like, does it require certain conditions to produce?
So yes, it does require certain conditions. Those conditions are pretty ubiquitous. So bacteria form biofilms in response to stress, but all environments are somewhat stressful. And so, as I mentioned in the beginning, up to 80% of bacteria in our environment are in these biofilms. Typically not individual, single microbe biofilms, which is what we work on in the lab, but with multiple different species. And sometimes actually also with archaea and eukaryotes as well. But these biofilms kind of just form in the environment, and they're very protective. So I only talked about one style of protection right here, about antibiotics, because that's kind of what we work on. But biofilms also prevent them against desiccation, predation by larger organisms such as amoeba, tons of other just environmental stresses and also nutrient starvation, because as I mentioned, the biofilm can be a place to store nutrients. So overall, microbes generally do form these all around us. I think I answered that question.
Yeah, I think that makes sense. So the next question came from Gary, and it was, are components of the biofilm matrix immunological targets?
Yes. So there's actually really cool work about this. MedImmune has, so let's back up a little bit. So people with cystic fibrosis get Pseudomonas aeruginosa infections. These infections are aggregates in the lung. And so MedImmune has actually used the blood of such people to isolate antibodies against Pseudomonas. And what they see is that one of the major antibodies that they make is against one of the exopolysaccharides that we know is in the biofilm matrix. So yes, they are immunologic.
Cool. So Wake One asks, we have two biofilm systems to treat perchlorate of groundwater, I hope I'm saying that right, for O&M. The balance among effectiveness, bioaccumulation, sludge volume is very important. Is it possible to get optimal conditions by controlling the components of the biofilm? And it looks like they clarified that two systems are fluidized bed bioreactor and hydrogen-based permeable film reactor.
Yeah, so theoretically, yes. So I think this requires a little bit more work than where we're at currently, because the biofilms for wastewater treatment are quite complex and aren't as well studied at a molecular level. But theoretically, so for instance, having too much biofilm is also bad in that case, because then they start floating. But the simple answer is yes, that in theory you could modulate how much biofilm is produced. I think that we're a little bit far from that currently, but that is definitely an application.
Awesome. Next question was, do the bacteria interact with free glucose?
So bacteria can take up glucose as a source of nutrients. Pseudomonas is not their preferred nutrient source. They prefer eating like short peptides, so small proteins, instead, and other sources of carbon, but they can take up glucose from their environment, if that's the question.
Okay. Another question from Gary. Do you think that enzymes could be engineered to specifically degrade biofilm matrix proteins or polysaccharides to be used as therapeutics?
Yes, so there's people doing this. I showed you that example of the 3 roles that matrix components has, and one of them was the degrading situation. Though the enzyme that's actually in that is actually produced by bacteria. Cleaving of exopolysaccharides, you might imagine, is actually important to biofilm formation, because you kind of need to break stuff so that you can kind of get bigger. And so if you just add the enzymes that the microbe inherently makes at a very high concentration to the biofilm, they do, so if you grow up a biofilm and then add it, like I showed you on the slide, the biofilm will degrade, like they'll just essentially wash off. The individual cells will, well, not individual cells, but the biofilm will essentially disintegrate. However, that does lead to now a lot of individual microbes in its environment. So whether that's good or bad for infection gets a little complicated. It would make them more susceptible to antibiotics. So if you did it at the same time as adding antibiotics, that might be useful. And people are working on this.
Okay. So Evan would like to know, is antibacterial resistance in biofilm due to the fact that the bacteria are slow growers, stationary phase, or an inability to penetrate?
So all of the above. So there's multiple reasons why in biofilms, the bacteria are more tolerant to antimicrobials. So one of them is, as mentioned in the question, that there's what we refer to as persisters, so cells that are in a different state where they're not growing. Physiologically, they're different. Genetically, they're the same as everybody else. And so those don't get hit by antibiotics as well, and slow growth in general. So as I mentioned in the beginning, most antibiotics target free-swimming bacteria that are actively growing. So most of our antibiotics kind of target this group, because our antibiotics come from microbes kind of fighting each other. That's how we find them. And so we have very few antibiotics. Well, we have some, but not very many antibiotics that actually kill bacteria that are not actively growing. The inability to penetrate is a problem for certain antibiotics. As I mentioned, these very positively charged antibiotics have this problem. In addition, these components of the matrix that are very negatively charged have been known to actually, well, I guess, been seen to sop up not just the antibiotic I showed, but also other antimicrobials that are positively charged, such as the ones that our immune system makes. So that's another element. In addition, there's just general heterogeneity. So the cells are not uniform in the biofilm. And so some of them are slow growing, some of them are faster growing, but there's also all sorts of other gradients in that biofilm in terms of oxygen, toxic products, and nutrients that affect the response of the bacteria, that affects their ability to respond, well, their tolerance to levels of antibiotics.
Okay. So the last question that I can see is, can you use biofilm to cultivate insulin from bacteria, similar to the way to cultivate toxins from botulism?
So insulin is not made in bacteria inherently. Insulin's a little complicated because it's the protein is just not like one easy protein to make. There's enzymes in our cells that process it. So theoretically, potentially, you could do that. I'm not sure if anybody's following up on that, since there's other ways to make insulin synthetically that obviously work now.
Okay. It looks like Evan had a follow-up. Could you reiterate the importance of extracellular DNA in biofilms?
Sure. Extracellular DNA provides structure. So that's been shown previously by Cynthia Church. So if you degrade the DNA in the biofilm, the extracellular biofilm, the biofilm will degrade. It also provides protective functions. So you can think of DNA as a long string of negative charge. Another way of thinking about it, because it has these phosphates on the DNA backbone. And these negative charges can interact with a lot of things. So whether that be antimicrobials that are positively charged, or ions, so they can actually be a way to sop up like cations from the environment. So let's say, iron is a very limiting source, and so it is a 2 to 3 plus depending on the state, and that could also bind to the negative charge. So it could be actually a way to hold on to positively charged compounds. So that's structure, that's nutrient source. In terms of protection, we think in the case of the image I showed you where the antibiotic is trapped around the outside, that is actually due to DNA interactions. We tried many ways to try to show this, but if we remove the DNA, the biofilm falls apart. So it's kind of difficult to kind of pin down. So all three, eDNA does lots of things in the biofilm.
Awesome. Well, I don't see any other questions. So thank you, and we've hit our time. So thank you so much again, Dr. Tseng, for your presentation. We really appreciate it. We appreciate you taking time out of your day, out of your research schedule. This was great. Thank you so much.
Yeah, thank you for attending, and thank you for your questions. And we'll see you next time for, I forgot her name, the woman from math.
Yeah, Dr. Neda.
Yes.
Yeah. Thanks everyone again for coming, and please join us next month for Dr. Neda. See you soon.